Hello all, welcome to Lesson 6 of our Unit 2 on DNA and genetics. This lesson we finally dive into the world of genetics. So let's begin. We're going to start out with our Geek of the Week. This is Gregor Mendel, born in 1822, died in 1884. He is considered to be the father of modern genetics. So you'll also hear us refer to his time in a lot of this section as Mendelian genetics, because after all, his name is Mendel. He was a brilliant student, but he had no funds for school, so he had to teach himself pretty much trial and error, had very little formal education. He was a teacher and a monk and a gardener. Okay, so like many people of his age or in his time, he wore many different hats, had many different professions going on at the same time. His most famous experiment was involving pea pods, so utilizing both his teacher section and his gardener section, he was able to sort of put the framework, if you will, together for our modern genetics. So his experiments was really the first work to show how traits are inherited. He wrote a paper called Experiments with Plant Hybrids. Unfortunately, his work was not really acknowledged as true or as correct for 34 years, which was well after he died. So he did his work. He made this fantastic study, moved the human race far above and well into an advancement period, and he got no credit for it until well after he was deceased. So that was Gregor Mendel. So DNA. We've talked a lot about DNA so far, and really beyond its nuts and bolts, which is what we were talking about when we talked about nucleotides. Okay, those were the physical structures of DNA. Really, if you think about it on a metaphor, DNA is a very large cookbook for us. Okay, it contains thousands and thousands of recipes that make us who we are. So you could have a popular recipe for an apple pie, but depending on who the baker is, your apple pie is going to come out very individualized. Same thing for DNA. DNA has the same basic recipes, but depending on which genes get expressed, makes up our own individuality. So if you were thinking again of DNA as a cookbook, the name of that cookbook would be genome. So all of the recipes, all of the genes that are contained within DNA is considered to be the genome. Now our human genome is 46 chromosomes long, and I'm pretty sure you guys know by now that we have 46 chromosomes. Each individual recipe or page of the cookbook is called a gene. All right, so green eyes would be an example of a gene. Brown hair would be another one. Okay, so those are genes. Some genes have natural variations, and those variations are called alleles. So for example, going back with our eye color, not everyone has green eyes. Some people have blue eyes. Some people have brown eyes. Okay, and some people have green eyes. Each human has 2 alleles for each gene. Okay, so 2 alleles for each gene. And remember, alleles are variations. So they have potential to be one or the other, okay, of the gene. And this will become more clear as we go along this lecture. So just remember, for this time, the variations of genes are called alleles. So whether you're blue eyes or brown eyes or green eyes, that is the allele of your gene or variation of it. Diving in again more into alleles. So the variations of genes. So alleles could be eye color, hair color, etc. And remember, every gene has 2 alleles. So if we look at homologous chromosomes, so if this were a karyotype, these guys would be, let's say, chromosome 1. Okay, so they would look like we had a chromosome here, and the number 1. Okay, that's basically what you're looking at here. Homologous chromosomes mean the same chromosome. So we know that they're in the same set. So they're probably both in just as our example. They're both in set 1 of our 23 homologous pairs of chromosomes. On these particular chromosomes exists the alleles for eye color. The left chromosome has the blue allele, and the right chromosome has the brown allele. Okay? Put these two together, these two alleles, and we'll get one gene that gets expressed. Okay? So the question is, well, when are they different, and how do you choose which one to use? And I know we're going to get into this in a little bit, but I want it to hit right now as just an intro. We know that we have two alleles, so each chromosome has their own allele of the same gene, and they could be written with either capital letters or little letters. We have dominant alleles and recessive alleles. In the case of brown and blue for eye color, brown is our dominant allele, and blue is our recessive allele. So just like the name implies, dominant means it's going to be expressed. So if we have one allele for brown eye color and one allele for blue eye color, the individual's phenotype, and we'll get into what that is in a second, but the individual's phenotype will be brown, because remember, the brown allele is dominant over the blue. So how exactly do we choose? Here's some expansion on this. The dominant alleles are the ones that are expressed even if there is only one copy of that allele, and they're usually shown as capital letters. So for the example we just talked about, the brown eyes versus the blue eyes, brown is capital because it's dominant. If we had a homozygous, which is the right term for this, if we had homozygous brown for a gene, each allele would contain the brown allele, written as a capital letter. So if we had, remember, two chromosomes giving the same gene but different alleles, one allele is brown, the other allele is brown, we have a big B, we get brown. Okay? In the case of the example you saw on the previous slide, where we had one allele on the chromosome for eye color as blue and one for brown, we would write it as big letter, so capital letter for the dominant allele brown, and lowercase letter or small letter for the recessive trait, the recessive allele of blue. And as long as you have one capital letter in this is called a genotype, as long as you have one capital letter, the dominant allele is going to show through as our phenotype. And again, we'll talk about that in just a minute as well. So dominant is going to show itself. So in our case, it would be brown eye color. Dominant would show itself as long as you have at least one capital B. If you have two capital B's, you're definitely getting it as well. The recessive allele is only going to show as lowercase letters. So for blue eye color to show, we need two lowercase b's for our genotype. Genotype. Okay? And that's what this thingy is called. When you write out the letters, it's called a genotype. So if you have two alleles of the recessive gene, then you get blue, for our example. Okay? So that is that. So again, genotype versus phenotype. Genotype are the two alleles on your chromosome. So for eye color, either dominant, homozygous, so two big B's, heterozygous, dominant, one big B, one little b, or two recessive alleles, which are little b, little b. Okay, when we write out the letters, physically writing out what I just wrote out, the letters, that is our genotype of our alleles. Okay, the phenotype is what actually gets expressed, what I can look at you. So if you have brown eyes, I'm either looking at two big B alleles or one big B and one little b allele that's made up of your eye color gene. Okay, if you have blue eyes, then I'm looking at two recessive alleles for blue eye color. Okay, so genotype is the physical written out letter format of my alleles, and then the actual color is the phenotype, what I'm looking at, what you can physically see. So phenotypes are things like eye color, height, weight, hair color, length, physical traits. Genotypes are the actual letter makeups of the alleles. So, genotypes are big B, and big B, little b, both have the same phenotype, right? Because remember, I said the rule is, as long as there's one big letter or capital letter in our genotype, the dominant gene will express. So how do you tell the difference between homozygous dominant and heterozygous dominant? And again, that's where we have homozygous, homo meaning same, homozygous, the same letter, heterozygous, different. Okay? Homozygous, same letter, same capitalization, heterozygous, different capitalization. So let's go through this real quick. Tall plants, which we'll represent with the dominant allele, capital T, and short plants are recessive, so they get the recessive letter. Now, as a trick, kind of keep in mind that whatever the dominant allele is, that's the capital letter that you're going with. So tall starts with T, it's dominant, we go with capital T. So then short would be the recessive trait, we just use the lowercase letter of the dominant one. That's why we use lowercase t instead of a lowercase s. So you're always going to have the same letter for your genotypes, okay? And that letter will come from the dominant gene. So for homozygous dominant of tall plants, I'm going to get one big T, two big T's. Same capitalization. Homo means same. Okay? Pure breed recessive means I have two lowercase t's because lowercase t represents recessive. And for me to see recessive as a phenotype, I have to have both alleles be lowercase. And heterozygous dominant is one big, one little. Okay? Hetero is different capitalization. Hopefully that makes sense. Hopefully you've got enough information now to complete all of the questions that are on your homework. I know that this homework is a little bit different as they do not completely follow the video lecture, but again, you should be able to use the information in this video lecture and transfer it to be able to apply it to your homework assignment. And so I don't forget, your video secret number for this lesson is going to be 71. 71 is your video secret number. Thanks so much for watching. I hope you learned a lot. If you have any questions, please write them down or email me them, and I'll be happy to answer them during class. Thanks so much.